always go through this sickness and all those. There's no such a thing as a perfect health. But though, if you're immunocompromised and if you're hospitalized, there you go. So you are prone to fungal infections. And some of the fungi are also opportunistic, like bacteria. If you give them an opportunity, right, they're gonna make use of it. And the term, if you remember, the bacteria, I told you that in your book there is a glossary for the terms that you just have to memorize. You know, we talked about infection, disease, colonization, parasites, all these kind of medical terminology. So there's one term called morbidity, which is like your health condition, how sick you are, how morbid you are, you know, as compared to mortality, which means the data that people die. So people become sick, there's a morbidity data by CDC. If people die, the mortality data. So higher the morbidity, higher will be the mortality. And also keep in mind that if you look at the bacteria, viruses, we discussed that the only way for them to go into us is via skin or mucous membrane. So it depends upon how much invasive they are. If they are invasive, if they're non-invasive, they're going to stay on your skin. If they're going to go deeper, they are invasive, right? And it can go and penetrate into blood and tissue. Bacteria, viruses, when we talked about viruses, we talked about their characteristics, how adherent they are, how nasty they are, how pathogenic they are, right? And the more deeper they go, the more difficult for us is to treat them. Like for example, if you have fungal infection, I'm going to give you a lotion. But if you have fungal infection of the liver, for example, or brain, then lotion is not going to work. Right? So this is called invasiveness. But I think the message is that there are no non-pathogenic fungi for all practical purposes. This means they are always pathogenic. They look colorful, they look beautiful, they're very, kind of playful, but they are not. The other important thing was that if you remember my story when I told about the viral infection, the first guy who noticed that in Atlanta, one of the pharmacists, that she was saying that this fungi should have been taken care by the immune system. Why is it that this particular person is taking repeatedly fungal drug? So that kind of raised a question mark. So why? Because you are immunocompromised, and then at that time AIDS. So AIDS is going to present as resistant fungal infections. Right? And then the other important thing is sometime in hospital, we kind of poke into your holes, right? So all those holes that the body has, they have this, catheters there, scopes there. So what you're doing is that you're introducing bacteria, viruses, and fungi there. So they have to be properly decontaminated or sterilized. But it's difficult, because one of the reasons is that all our bodily holes are already having tons of bacteria and viruses there. And if you poke a hole, you kind of take them in. So that is, the more invasive a surgical procedure is, the higher the chances are that you are going to get sick. Okay, now a little bit of taxonomy structure. They have their own kingdom. I already told you they are eukaryotic. And these are some of the key points that you have to remember without going in further detail, that they are distinguished from other eukaryotes. Yeah.
What makes transplant patients, what makes them immunocompromised?
Transplant patient?
Yeah, are they just talking about any transplant?
Because when you have to transplant an organ, you give steroids so that the immune system doesn't attack. So you give steroids to suppress your immunity so that your transplant is held. Does that make sense? Because if you're going to transplant without doing the screening, your body is going to reject that. Body doesn't want, just like blood transfusion reaction. So in order to stop that, we give steroids. So that's one of the reasons. So the important thing is that they have a cell wall which has a chitin and glucan. And the most important thing, very commonly, which is asked, is that they have ergosterol as compared to cholesterol. So these are some, because the idea is that they are eukaryotic, but a little bit different. So we still have a window of opportunity to go after them, and not on your own, not your own cells. Right? So this is a very important conceptual thing, so that you understand. So if you look at the structure, for example, this is a typical structure over here, and you can see that they have a cell membrane, okay, and they have nucleus, pretty much the same that we have, but they also have a cell wall, which is composed of chitin and glucan, and it gives them rigidity, right? And then again, we have cholesterol, but in the cell membrane, if you look at the bilipid membrane in human, we have cholesterol, but they have ergosterol. So that's good. So why do we need a structure? Because we're gonna go after them, not our own cells, and go and design those drugs which are going to target these compounds and not our body. That goes for everything. So you cannot use a drug to kill a microbe and end up killing yourself. Okay. Then again, a little bit more details about the capsule. Like all capsules, in this way, they are important. I'm sorry, because it kind of protects them. So your cells cannot engulf them. So it is an anti-phagocytic. And then all the capsules in bacteria also are virulence vectors. The harder the capsule, the difficult for us to kind of break. So just imagine that if your drug is going to go and act on the cell membrane, and the cell wall is outside, and outside cell wall is a capsule, you have to penetrate the capsule before so your drug has to go. Okay. So I'm going to talk about a little bit later, some of the other work. And if you look at the cell wall, like all the cell walls, and these are like structural things, all the cell walls are antigenic. What it means is that your immune system is going to see that. So all the external, outermost thing is going to be seen by your immune system. We call it antigenic. And if you look at the real structure, they are mostly polysaccharides and hexose and hexamine polymers. And why do we need to know these details? Because we're going to go and drug. So we're going to dismantle them. So we should know what the structure contains. And if you compare them, you can see that polysaccharide carbs are more than proteins and glycoproteins. Okay? And as with bacteria, the outermost is like eggshell. So eggshell keeps the form of egg. Right? It holds everything inside. And it gives them a shape, rigidity, strength, and protection from osmotic shock. So this means that if you just want to give, like for example, if you're going to put the egg into a hypertonic saline, nothing's going to go in because it's protected. So it is very important for us to know, as we remember, when we talked about the bacteria, we talked about some of the bacteria which have a outer layer which protects them, and it's very difficult for the bacteria to go in. So this is like a biofilm. So they make like an outermost covering, like it's raining outside, and you put like raincoat that kind of protects your clothes. So it's the outermost covering that's going to protect them for any other things that you want to do to kill them. Okay. And I think, is anybody using clickers right now? Okay. Any instructor using clickers? Okay. So there are some changes that we are making as we're moving along, you know. So I think probably most likely I talked to Dr. Walkina, the higher administration want us to use clickers. So clicker was like, I'm gonna post a question, right? And you will have this kind of this machine or your cell phone, you press it and your answers will come. I don't know how many of you have done. So these were designed when the question, the lectures, these are the lectures, they were designed in a time when we used to have clickers. So that records your data. You just imagine saving time instead of me asking you to write an exam. And so they keep on coming with experimentation with new ways. So we started with the clicker, we went to flipped classroom, and now I hear again from the chair that we are going to go back to clicker. So these are some of the questions which are clicker based, but I'll just go through with you anyway. So this is like a question with, you know, select the incorrect statement about the size of fungi. So these are the choices that you have. And then again, I'll ask you to press the number by the clicker. And then again, the correct answer is incorrect. It's not the size of the virus. The virus is smaller than that. So this is like questions that we have. Okay. Now, without going into detail, but keep that in mind. If those of you, and it's very difficult for instructors to teach you the whole microbiome. I can't teach you bacteria, virology in one lecture because I have to break it up. Because it's going to be too content heavy, right? So that's why I keep on saying it's better for you to be ahead of me, so that you know where things are coming up. So all these microbes, they have features like the structure, which is important, how big they are, how small they are, about the sterilization thing, how can they pass membrane, they cannot pass membrane, and then again, what are their biochemical characteristics, what are their molecular characteristics, because this is going to be the basis of the drugs, you know, where the drugs is going to act. But keep that in mind for at least this class, because I'm just going to start with a very basic one. Fungi can be, they are usually multicellular, but they can be unicellular. Okay? And in normal, our day-to-day life, I see we talked about yeast. So yeast is a fungi. In food, we use yeast. And I think at this level, I'll wait till DAS 7 and DAS 8 is in P4 probably. So you have a couple of years to kind of digest that. But right now, I'm going to lump them together into yeast and molds. This is very oversimplification, but since you know you are in P1, so I'm going to. Now, having said that, some of the bacteria may have a morphology which is we call dimorphic. Keep that in mind that exceptions are everywhere. I say this is a DNA virus, RNA virus, but don't take it literally. You know, what we are saying is that human biology has heterogeneity. We teach you to make things simple, but it's not that simple. Okay, so I just said they're either yeast and mold, but now I'm saying it, sometimes they're both. And interestingly enough, those who cause diseases are dimorphic. So let's go for the yeast, and you can see yeast are unicellular. If you look at it's just like budding, if you see like potatoes, or say they kind of bud out. So it's like mother-daughter cell kind of distribution. And sometimes they kind of branch out, right? And they look that they are multicellular, but they're not. So if you look under the microscope, for example, over here, you know, on the top one, then you can see that they divide into two, right? On the top, this is a typical division. But when they bud out, right, and they form a longer structure, it looks like multicellular, but it is not. So this is a branching, hyphae is a branching, but it's a pseudo branch. So it's like, we call it, what do you call, Pseudo means pseudo, it's not true. And you can see we have pseudo hyphae. And many a time what we do is that, this was like, you know, when I was teaching in biology, who's teaching 101? Have you?
Probably Daniels or McDonald.
Daniels. So 101 biology, so they want you to take a fungi and look under the microscope. This is one of the 101 biology at our, so what you will see all these structures. So what they do is, the clinical, they take the scraping of your fungal infection and look it under that. So this is how they look. And these are the terms that they use. So this is a yeast, A, and B is a mold. But you can see that if you're not smart enough to figure out, which most of us are not, because these are very specialized slides. So we may take a yeast as a mold, because under the microscope it's very difficult to tell. You see? But they have cells. Right? The terms that we use for them both are hyphae, right? And the technical terms in mycology, as you see, every subject has a jargon. Like bacteria has its own jargon, virus has its own jargon. So the jargon over here is hyphae, right? And conidia. And blast is like blast. You'll see that these terms are repeated, conidia over here, and then the cells. So these are some of the terms, and if you look at these structures, they may appear as a multicellular. So you can see from here, molds usually are multicellular as compared to, I mean, that's like a very simple classification. And I already told you multicellular, and then the terms are, one term I didn't mention was mycelium. So it's like a mesh, it's like a mesh. Like for example, you know, if it's like one straight and just kind of mesh it up. You know? Like for example, the back of it is just like a mesh. This is mesh. This surgically we call it mesh. This is mesh, it is called mesh. So that mesh-like structure or matrix is called mycelium. They are interconnected because they're budding out. So mycelium, these are the terms, septate, mycelium. You will see, and I did one of the I think it was when another review is on the Moodle. Apart from lecture, I put like review 1, review 2, review 3. So I find that that is giving you a, what do you call, a bird's eye view of the whole structure. It's difficult for me to do it on an individual PowerPoint slide, but if you look at a big picture, big placard, you will see the divisions and divisions and divisions. So that's another way of learning. We call it concept maps. Some of the students in the past have made it, right? And I used to ask them to share it, but I don't think that's needed, because you can work it on your own, but they're available. I just added the one that I like, and I for my own memorization, and this is what it is. So this is the most important question that I'm going to ask you, and everybody will ask you. What is the difference between mold and yeast? Right? Simple is the yeast are single cell and produced by budding. Mold, so I usually give a mnemonic, M for mold, M for multicellular. It's as simple as that. So you can make your own. So you don't mess this up, because if you mess at this level, then you're in trouble. The other problem with the fungi are that fungi look like plants, right? And you can see from here. So they are, they look like plants. So these are hyphae. So keep that in mind that you take in fungi by respiration. So if you are walking a path, a park, and most of the diseases are acquired respiratory, and you blow on something, and these are airborne. So you can see from here, if this breaks open, so all these kind of things which are there, they will be airborne. Okay? And then you breathe them in. And then they start multiplying. So this is like pathogenesis. And they come into, you say aerial means in the air, hyphae, and they are, the reproductive cycle in this case is asexual, right? If you look at the two important thing I think you have to remember is that, this is gonna get a little bit harder, but there are diseases associated with that, and I know for sure they teach you in P4. So one of them is, you can see from here, aspergillus. Aspergillus. This is a disease, aspergillus, right? The moment you hit aspergillus, you will see it's a fungi. And the other is zygomycetes. Zygomycetes. And these are the typical examples. You see zygomycetes and aspergillus. So if you, next time when you go and buy, or you can do it today, you go to Walgreens CVS and pick up a antifungal cream, whether it's a for lotion or vaginal suppository or anything of that sort, which has fungal infection, you will see that the names of these fungi will be written. And some of them even have the name on the brand. Okay? So this is something of memorization. You remember I told you problem with microbiology is too much memorization. And you have to do it in time, otherwise it is very difficult for you. Okay? So this is the septate hyphae, and both of them, you can see this was covered, right? And it has to break apart to spread, as compared to this one, which is going to explode out by the air or breathe or anything. And both of them are there to cause diseases. But this is an example of a typical asexual production of fungi. So this is another very common question asked. You can see the difference between fungi and plants, because they look like plants. And these are the questions from the chance book. He's going to say like this on the left hand side, say this and answer, this and answer. So I mean, I and my students in the past have liked this. So this is the most important thing. The characteristic of the plants is that they have chloroplast and they have photosynthetic energy producing mechanism. These are plants as compared to fungi who do not have that. So there is no confusion. It's a simple biology, plant biology. They teach you at 101 level that chloroplast and the photosynthesis are there. As compared to fungi, they look like plants. Okay. The other important thing, if you look at bacteriology, is that bacteria, they divide, they form colonies. Viruses don't form colonies. If you look at, so we grow bacteria on a plate, and you will see they kind of form colonies, which is called colony forming units, CFUs. Okay. Now, these are the characteristics of the colonies of the fungi. So if you, even if you see a rotten, piece of bread, you will see, kind of a spread out. Some of them, if you look at, they are filamentous visually. Some of them are hairy, and some of them are woolly. So these are the characteristics. So if you see that, it's just like mold in the home. You see, wherever there moisture is, your washrooms, wherever water, you see this black thing growing over there in the corner, right? And you inhale them, and then again, mold is a big issue for housing because you breathe them in. And they are aerial, so they come in the area. That's why all these vents are tested for molds. If they're not been testing, we're gonna all have fungal infections. That's why if you go to rundown houses, you can't buy them because somebody has to test a house infection for mold. So mold is one of the big problems that we have. Okay, the other important thing that you can see, these are some technical terms that you need to see, but I think I'm not gonna spend much time on that, but I think using your common sense, and I said that in viruses as well, if you know the structure of basement membrane of alveoli, basement membrane of kidneys, and the structure and sizes of the pores there. So you can imagine how small the size of a fungi have to cross them. So if you inhale, but they're too big, the respiratory is 2 micrometer. It's not going to go in. It has to be less than 2 micrometer to cross and invade, invasiveness. So invasiveness is something of size. Smaller the size, and you can see from here, the fungi, big plants, but the little conidia, very small, and you don't see them, and you're gonna inhale. And they can even cross your blood-brain barrier. So there in physiology, there are 3 important things that you have to know all the time. Is the structure of blood-brain barrier, glomerular filtration for the kidneys, respiratory membrane for that, because these are the 3 places where microorganisms are going to cross. And that's why size matters. Okay. But there's one catch to it. One catch to it is that the size is small, right? But you have infection, and inflamed is going to get bigger. So inflammation is going to increase the size and then let more microorganisms come in. Okay. Now for the fungal metabolism, most of them are aerobic respiration, and some of them also ferment. And you see yeast, you use yeast as a fermentation. And then again, some of them are strict anaerobes. So these are the terms that we use in bacteriology, something that you have to memorize. You have to know what are aerobes, what are anaerobes, what are strict anaerobes, because there's like 3-4 of them, and there are a lot of mnemonics for that, but you have to keep in mind that they have aerobic respiration. Mostly, they need oxygen. But some of them are anaerobe. They can go without oxygen as well. Okay. Now again, the clicker type question. This is again will come. In the past we used to have, but we changed it, and I'm just giving you a heads up. It's going to come back again. I don't know when. So it's like most fungi are anaerobes. Some can grow under anaerobic condition, few are anaerobes. Anaerobes are not human pathogens. What's the correct answer? All of the above. It was easy. Okay. Now, as far as metabolism is concerned, and keep that in mind, fungi are used in food industry. Right now, I haven't seen any food product that doesn't have citric acid. Right? If you look at the ingredients, all these packed stuff, you name it. Where is it coming from? They're coming from fungi. So we use fungi to produce them. So, and we also need to keep in mind that even penicillin is a product of fungi. So the top drug that we have, antibiotic, penicillin, it is a fungus. Okay. The difference between bacteria and fungi are that they are very slow. It's like yeast. It takes a time. Bacteria are faster. Okay. Now this is where your memory test is going to come up, and I think I tried to be as simple as that, but I think there's nothing less. I can go for anything less because they all have diseases, and they're gonna. You need to know them, and you need to know the treatment as well. I'm not keen on treatment except like very basis, but eventually down the line you have to know that. I'm gonna help you with some of the things just to make you understand. So you can see one of the things that you will see, all of them are ending with mycetes. All of them are ending with mycetes. So it makes your life easy, they are all fungi. And zygo, I already told you, archaea, basido, hemi, and euco. And then eventually I'm going to talk about the diseases that they cause. Okay? But I think there are 5 major classes which are human disease. And So these are this is like Staphylococcus cocci, aureus, and then what disease is caused is different. The name of the micro and the disease may be different. Sometimes it's like Mycobacterium tuberculosis causes tuberculosis. Mycobacterium bovis causes, so it makes, sometimes it's not. So this is how I already talked about that the most important thing is that these 5 classes over here, they produce both sexual and asexual, right? And without going in detail, I think you pretty much know what is the difference between asexual is usually mitosis, this is biology 101. And sexual, which means meiosis. Same basic biology 101, right? And it exists in both of these forms, and we give them a different name clinically, right? But you can see from here, the fungi in the 5 classes that I just talked about, they both have sexual and asexual spores, so they can produce both these. So when you take a sample, for example, there's another thing that when you take a clinical sample, right? So keep that in mind that many a time we are able to pick the asexual form. Right? It doesn't mean that they are asexual, because for sexual form of the fungi, you have to give them a specialized condition in the lab. So you take them out for asexual, put them on a plate or media, and then they start producing in a sexual way. So there's a clinical specimen collection. We used to have a separate lab for that, but keep that in mind that you collect a sample. I'll give you an example. For example, maybe in the clinical part, I don't run ahead of my lecture, is that whatever sample you take, right, so it's usually asexual. Doesn't mean that it is only asexual. You have to grow it in a medial to figure out what's going on. Now, we are only concerned with human disease. So there you can see from there are thousands and thousands kind of fungi. 200 are known by human disease, but we just need to know 5. So that makes your life easy. That out of 200, unless you want to go for a mycology expert, or you want to be specialist in fungal infections. And guess what? It's one of the very common lucrative job in cosmetics. Because cosmetics, the major issue is fungal infections. Okay? So these are the very basic rote memory things that I mentioned about, right? Now let's jump to the human one, because I'm going to go more for clinically, and these are the 5 important, I would say, fungi which called human mycosis. We're not talking about animal, we're not talking about all of that. So human mycosis, and all you need to know is your skin. So you have to go back and look at the skin structure, dermatology. That will make your life easy. So if the fungi are on the superficial, right? So your skin anatomy will tell you what does superficial mean. I will also talk about that. The more invasive it is, like for example, if this is the top of the skin, like this is like superficial, right? And it becomes, from superficial it goes to cutaneous, then subcutaneous underneath, and then endemic, and then opportunistic. So it's like depending on which layer it caused. And this is what you see in clinical practice. Everybody's worried about nails, everybody's worried about the hair, everybody's worried about skin. So that's why I say cosmetic industry. So hair and nail are extensions of skin. Keep that in mind. That's what I told you in physiology, that they are part of the skin. And the biggest industry right now, even bigger than pharmaceutical, you may become sick or you may not become sick, But everybody in the world, male, female, is worried about his or her skin, nails, and hair. That's why I said the biggest industry out there. And All of them have fungal infections. Okay? So what is the challenge? The challenge is, this is the challenge. Since they are eukaryotic, they are biochemically similar to human hosts, it is difficult to develop chemotherapeutic agent that will destroy the pathogenic fungus and not harm the patient. That's the challenge. Okay. As far as the classification is concerned, you can see, I just told you, it depends upon which part of the body is affected. Right? Cutaneous means skin, subcutaneous means under the skin, systemic means it has gone past the skin into your body, into your system. And the rule for that is, if something's in the blood, so it will go to wherever the blood takes it. Okay. How do we diagnose that? So what we do is that these are again 4 important tests that are available. One is that you just take the scraping and look under the microscope, which is not that easy as you saw. Then you can culture. And I told you, why do we need to culture? Is because to differentiate between asexual and sexual, right? You can do the serology antibody test, or you can do DNA probes. So these are all things available. But the most important thing that you need to know for the culture is called Sabouraud's agar. Right? And what we do is that we put a little bit of antibiotics there so that we can grow fungi. Right? So if you are given a clinical case and it says Sabouraud's agar, we are talking about fungi. This is how it looks like. These are colonies. These are like colonies. Okay, and I'll stop here and take questions. So I'm going to go a little bit slow because there are a lot of memorization points and a lot of stuffs available. And any questions? And whatever I've said is, I told you, it's pretty much the same lecture. Is pretty much exactly the same slides. So in my YouTube and the podcast, that's all basic, back to basic. All the rest is your memorization. And I told you, if you look at Moodle, you will. Those of you who want to go for the look at reviews, that gives you the flow charts. I am a visual person. I need to see a bigger picture. So whatever I like, I just put it up there for you. Any questions so far?
Does fungi produce oxygen?
Why would it produce oxygen if it doesn't have aerobic respiration?
During my microbiology class at my previous college, I was told that fungi produces oxygen.
In what sense?
And I asked them a question that
It's not a plant. It takes in is aerobic respiration. It needs oxygen because it's, if you remember, but as far as production is concerned, that's totally, because we talked about the biochemical like citric acid, ethanol, glycol. These are yeast, whatever is there. Any other questions and concerns? So I just gave you a very basic stuff, and I think I'm gonna limit myself to the human disease. And what I can give you, I would suggest to you, for example, if you don't have any questions, let me see. There are 5 lectures. Yeah, so this is what I want you to kind of at least, for when you come for the next lecture, you just kind of revise your physiology, this part. Because if you understand that, you will see, because the important thing is skin. Fungal infection is skin. That's what most of the time, skin. Right? So if you know the structure of that, you can see from here, the structure, you'll see epidermis, that's where the first part of the fungal infection, then you see dermis and hypodermis, and then you can see all these kind of things in terms of problems that we have for skin. People care about skin. People care, and I can tell you right, so the companies are interested because you're gonna use skin ointment for rest of your life. You're gonna use nail problem rest of your life. You're gonna have hair problem rest of your life. The only thing that I'm gonna add will be more detailed clinical problems, which are a little bit rare, except vaginal discharge. So that's again number one cause for fungal infections. And that's why if you go for the over the counter, you will see skin ointment or treatment for fungal infection with vaginal discharge fungal infections. Very common. Any other questions or concerns? None? So we'll see you on Wednesday. So make sure that you read it up because I will spend less time on that. All right, Xavier, you good?
Yep.